So next, I'd like to introduce Dr. Wendy Chung, a clinical and molecular geneticist from Columbia University and a world leader in the diagnosis and treatment of rare diseases. I first met Wendy when my daughter Emily received her diagnosis. Our neurologist had ordered the genetic testing, and when the results came in, he said to me, "There's only one person to see, Dr. Wendy Chung." Dr. Chung wears many hats, and I'm pretty sure she doesn't sleep. In addition to her clinical practice, she runs her lab, the Chung Lab. She directs NIH-funded research programs in human genetics of autism, neurodevelopmental disorders, birth defects, and many other programs, and is the director of clinical research at the Simons Foundation's Autism Research Initiative. I'm not even touching on all her other qualifications and jobs and things that she's involved with. When we started the foundation, one of our first initiatives was to start a natural history study so we could better understand our disease. I reached out to Wendy for advice, and she said, "I can help you with that." Last year, Dr. Chung presented on her initial findings from the first 35 patients that had enrolled. We're so grateful for her support and are looking forward to her updated findings. And just so you know, if you have not enrolled in our natural history study, after hearing Dr. Chung talk, I know tonight you're going to sit up late in your hotel rooms and fill it out, because it's really one of the most valuable things that you can do. Thank you. And Dr. Chung is virtual, so here she is.
Thank you, Lisa. I'm sorry I couldn't be with you there in person, but I'm there with you in spirit. As Lisa said, it's really important, I'll try and explain why, but to be in the natural history study. Let me see if I can advance my slides. So the goal of the natural history study, and it's constantly improving, so we love to hear your feedback, but is ultimately, the ultimate goal is, as I think of it, the care until the cure, and it's both the current care and the future cure. So what I mean by that is the following. You've heard a little bit about it already this morning in terms of the heterogeneity or the differences, both in terms of the variants within the gene and the manifestations. And so there's heterogeneity within our community, and we're trying to understand really the diversity. And to a certain extent, as you're going to be hearing throughout the meeting, some of the science behind that in terms of how this channel functions within the brain. We also realize in a good way that things are changing over time. What I mean by that is that as some of you have figured out what I call life hacks, being able to think about what's working, either in terms of treatment strategies, medications, things to avoid, but as you're learning these things simply by trial and error in some cases, we want to disseminate those experiences and be able to hopefully help our next person down the line to be able to benefit from what we've learned. In addition, those of us who are doctors are doing something similar. We're looking at the medications that, again, we or other of our colleagues have been trying and seeing what's working better, what's maybe not working so well, what we can do in terms of management to help it make it better until, as you're going to be hearing, some brilliant people are going to be working towards the cure, working towards new treatments, new medications, maybe perhaps even things like gene editing or gene therapy someday to be able to take care of this in a more permanent way. I won't make any prognostication about if or when the cure is going to happen, but I will say that there are a lot of people working in a lot of brain disorders, not just CACNA1A, and I think we will be able to benefit from the things that they're learning. In addition, I'll say that things change over time. Things change over the life course. And as you're going to be seeing, we're mostly a young community. And so in terms of who's in the room, who's out there virtually, who you were seeing in terms of the videos, most folks that are represented within the registry I'm going to show you are young people, but that's not to say there aren't many, many more people out there who are not yet represented, who don't yet know they have the diagnosis. I would venture to say for every one person we know of currently, whether they're in the registry or whether they're known somewhere, I would bet there are at least 100 other people, maybe even 500 people out there around the world who don't even know that they have this diagnosis. And so part of what we need to be able to do is raise awareness. And I can say from an advocacy point of view, at least in the United States, I've been working hard in Congress to make sure that everyone can get access to that diagnostic testing. So, as Lisa had said, if you do decide to stay up late tonight, here I'll put it at the end also as well, but here are the ways to be able to start the study. And again, I'm always open for feedback because this is an iterative process. So you can email either Alexa or Sean, and these are their email addresses here, and it's relatively easy to do the first steps and sign up for the study. As I'll be showing you, we want to make sure everyone's one who thinks they belong in the CACNA1A community actually has a genetic variant that puts them in this community. It's very unusual, but I can say that there have been some circumstances where individuals have misunderstood the genetic test report. I'm not blaming anyone, but it's very complex gobbledygook, and sometimes there's something written in there which leads you to believe that this is the right diagnosis. And it's rare, but sometimes it's not. And I want to make sure every family gets to the right diagnosis, because ultimately you have to be lined up to get the right diagnosis, to get the right treatment. So that's step one. We ask for a copy of your genetic test report. Hopefully your doctors have given you that. If they haven't, we're glad to help you request a copy, but that's the first step. I will say that many people they've signed up for the registry, but they haven't yet gotten to that first step. And we don't want you to waste your time if you're in the wrong club. So that's kind of a gating feature for us. The next step is we do this all online. So you don't have to come to Columbia. You don't have to even be in the United States. We do this so that you can do this in your pajamas at 3 o'clock in the morning. I hope you're not up at 3 o'clock in the morning, but to be able to do this at your convenience. You can stop and come back if something comes up. And importantly, this is not one and done. So this is something where we will continue to follow individuals over their life course, because as I said, things change. I hope they change for the better. That is, that we learn, but we also are concerned in terms of as people go through different life stages, where it's adolescence, young adulthood, older adulthood, that there may be things that change that we need to keep an eye on, and again, learn from each other as we're doing it. Okay, so by the numbers, let me just tell you a little bit about our cohort. As I described, this is an age distribution. All of these slides will be available afterwards, by the way, so no one has to furiously make notes, including the scientists. All of this will be available to you. Right now, we have just shy of 100 individuals on whom I'm going to be telling you what they've shared with us so far. This is showing you just roughly the decade of life that they're in. And again, you can see that most individuals are below the age of 20. We do have a few young adults here in the decade of the 20s, and even fewer, though, that are up into the 30s, 40s, or 50s and 60s. As we divide them out, I do expect that this should be an equal opportunity genetic condition. And what I mean by that is that it should, biologically, the way we think of it is that it should affect males and females equally, equal opportunity in that way. And it should be relatively similar in frequency around the world. So we don't know this to be, for instance, Puerto Rican condition or a Chinese condition or a Norwegian condition. We should be seeing it equally around the world. To the extent that we don't see it equally, I think that represents differences in who has access to genetic care. But right now we're seeing, again, based on many families being in the United States, but I think they are around the world. So this is a complicated slide, and it's mostly for the scientists, but let me try and orient you a little bit. And I'll try and throw in a little bit of scientists, but I will say you're going to hear from many of our esteemed colleagues who think about electrophysiology and think about channel biology. It encodes one subunit of a calcium channel. So I think of channels as being holes in the cell membrane. So we all have cells in our body, and they have an envelope that keeps those cells sort of contained, and they have very small openings that allow different salts to be able to go in and out of them. And those salts coming in and out are electricity. And there are organs in which that electricity is extremely important. And the brain is one of those organs in which it's extremely important. The heart is another one, but for CACNA1A, it's especially the brain. Those holes allow regulated influx and efflux coming in and coming out of those ions. And it's very important in terms of keeping the electrical sort of connections in the brain working correctly. Now, this channel, someone mentioned earlier that it's a big gene, and that's true. It actually is a very big gene. And because of that, there are multiple places where there can be mischief within this channel. This channel is what we call heterodimer, or rather a heteromultimer. That is, it's composed of multiple subunits that all come together. And this is meant to show you the full gene. So it starts way over here and it ends way over here, and it curves in and out of this cell membrane. And within this, there's some complexities to this. So this forms what we call, and within this, there are what we call 4 transmembrane repeats. So they're marked here, 1, 2, 3, and 4. And they're not exactly the same sequence, but they're 4 different repeat regions. Within that, there are some particular, what we call transmembrane different regions here, S1, S2, S3, S4, S5, and S6. And each of those are in these 4 transmembrane repeat regions. And so I've color-coded these so you can see approximately how those go. Each one of these dots that you're seeing here represents a different genetic variation that we know to be associated with some sort of neurological manifestation that we see. And I'll get to it in a second, but it's really a diversity of different types of manifestations that can include everything from episodic ataxia to hemiplegic migraines to epilepsy to developmental delay. I'm including all of those here. So I consider this kind of gene-centric, not necessarily based on the symptoms. And some individuals can have more than one symptom. Now, if you were to look at this just very quickly, you would see that the distribution of where the dots are not completely even throughout the gene or throughout these different domains. And that impression is in fact correct. We tend to see a clustering of the particular mutations or genetic variations in S4, as well in S5 and S6. And in particular, we know that that's the case because S4 has a region that contains some positively charged amino acids that really aren't happy when you start changing those charges or changing those amino acids. Those, in fact, are the regions that are the voltage centers within those channels. And all the gobbledygook, don't worry if you don't get it. S5 and S6 are the ones that line the channel conduction pore. So S5 and S6 here. And so you'll see that in different regions of S5 and S6, you tend to see more of those dots that are located. Okay, so that's a basic orientation to the channel. Now, I've also shown black and white and gray dots. So if you look at the colors of these, you'll see that they're in fact those 3 colors. And this is a key over here on the right. Now, these are based, for the scientists out there, these are based on using funkion as a prediction. Some of you are going to be hearing about actual electrophysiological studies to directly experimentally test, and there may be some discrepancies in what I've shown here. I will not say this is perfect, but I wanted to give you a best guess in terms of being able to do this computationally. Now, with this channel, this functions in terms of opening and closing, and it's very complicated, not just that it's always open or always closed, it's actually dynamic in terms of sometimes being open and sometimes being closed, and knowing some of those very specific dynamics of what triggers it being open and how long it's open. In a very broad way, and again, very broad, simplistic way, I've coded these for those that are in black to be what we call gain of function, or in other words, this channel staying open too long. In white being loss of function, or LOF, that channel is being more closed than it should be. And in some cases, particular genetic variants that we think just make the channel not be there, or at least one of the 2 copies of the channel not to be there. And that brings up an important point, that each of us have 2 copies of this gene, and that for most individuals that are in the community, we know that 1 copy of the gene is working just right, and it's one other copy of the gene, one of the 2 copies of the gene that has these types of genetic variants that I'm describing. For most of the individuals that are in the registry right now, we know that this genetic copy that has the variant actually started de novo. You've heard about this earlier today, but just to confirm, in our registry, most individuals, this was not passed down from the mother or father, but started brand new with the any individual. And just to reassure you as parents, there was nothing, especially you moms, nothing that you did or did not do during the pregnancy to cause this. This is just something that happens, and we know it happens in every single pregnancy to a certain extent. And in your child, it may have happened in this gene, and just by chance, we see this association. But all of our children have de novo genetic variants that are not present in either parent. Now, one other thing to orient you to is that when we see the distribution for most individuals in here, these are onesies. What I mean by that is unless you see a number in parentheses after the individual genetic variant, it is represented by one single person. As you can see, most of the genetic variants that we've seen so far are those onesies. They are unique. Again, it's not that I think they're the only ones in the world with this. I think there are other people with the same genetic variant. They simply aren't in the registry or don't know and haven't because they haven't been tested. However, there are certain places where we see the same genetic variation over and over again. And these are not because they're related. So it's not because they did ancestry.com and they figured out they were long lost cousins. They're completely independent in this, but yet that same position has happened over and over again, and oftentimes has come to clinical attention because it's been associated with more severe symptoms. Okay. I know that was a huge amount of information, so we're going to breathe for a second, and now we're going to get to the things that you probably care much more about. That last one was mostly for the scientist. With this, and you know this just based on your own experience, but to share it back with you, we know this largely is affecting the brain and behavior. And I will say that I think we have some ascertainment bias in terms of who gets genetically tested, so that many of the individuals in the registry right now have largely epilepsy and developmental delays, and at some point may be diagnosed with intellectual disabilities. Not everyone, but I do think we have overrepresentation of those individuals because that's oftentimes who doctors think about genetic testing. And again, it tends to be younger individuals who were diagnosed in an era when the genetic testing has been available and has been able to recognize this. So again, I think there are more people out there, many of whom are older and or have milder symptoms. But we tend to see the following, and hopefully this is something that resonates or feels like it's correct. We see individuals who are later in walking and talking, as you've been seeing from each other and in the videos, have problems in terms of coordination of movement, may seem clumsy, may be ataxic, may have problems with balance, but they're having mostly what I call above the shoulders problems. That is, the origin is a central nervous system or brain origin, even if it manifests in terms of the way the rest of the body is behaving. In addition to that, and sometimes it takes a while to see these as the individuals are getting older, we can also see some behavioral issues as well. We can see problems with being anxious. We can see problems with focus or attention. We can see problems with being sad or depression, and problems with obsessive compulsive disorder. Now, again, in terms of seeing the frequency, it is the majority of individuals who don't have these conditions, but for some individuals, and sometimes it's because of what people are able or not able to do, that they may be sad about those particular limitations. So it's easy to understand how these pieces can fit together. We've also been able to look at the brain imaging, or MRI, in many individuals. Not everyone, but about 84 individuals. And so I'm showing you, of those 84 individuals, what we see. For some individuals, we see nothing. About half of the people, their brains look, when we look at the scans, just like anyone else. On the other hand, there are some individuals, you were hearing earlier about a part of the brain in the back called the cerebellum. And in fact, the most common thing that we see is cerebellar atrophy, or the cerebellum being smaller. And with dysfunction of the cerebellum, as you were hearing, is where we have problems in terms of movement and coordination. There can also be problems not only with the cerebellum, but also with the rest of the brain as well. And in certain circumstances, we have seen problems, again, thankfully, not usually, but problems in terms of swelling or brain loss. This I realize is very small, and I know is going to be too small for you to read, but let me just tell you about it. And again, I will post the slides afterwards. You've been hearing as well about other things with the eyes. And so I just want to also endorse, and this is from a publication that we recently had summarizing all of the data that you can be able to see online. We do see those problems as well. So we see problems in terms of eye movement, all of the things that you've been hearing about earlier, but problems that are largely coming from the brain in terms of controlling eye movement. I just mainly wanted to endorse that we see that. We also see the problems in terms of hemiplegic migraines in about a third of individuals, and we see some systemic problems in terms of hypotonia or low muscle tone, as well as ataxia, both of those in about 3/4 or 75% of individuals. So again, just to endorse that those things that you've been hearing about, we've been seeing as well, and leads me to believe that the registry is accurate in terms of the data we're seeing. Okay, let me tell you something we did in the paper that I just alluded to. And again, we'll make sure that everyone has access to that paper if they like. This was based on my own personal experience as a clinician. I tried to come up with an overall sort of my gestalt over whether I thought some individuals had heavier burdens, whereas other individuals might have had slightly lighter burdens. And again, I'm not dismissing the burdens for anyone. I take all symptoms seriously, but I was doing this to try and understand if there might be any association between the specific variant I was showing you in the channel before and the manifestations. And I was doing this because, as you can see, many individuals are the first person we've seen with that genetic variant. And one of the questions that people come to me as a doctor with is they say, what should I expect? I have a 2 or 3-year-old who was just diagnosed with this condition. I don't know anyone else in the world who has that same genetic variant. Can you help me think about what to expect? So we've been trying to model this, and we've been trying, as many of the other scientists you're going to hear about, trying to predict the ion function and trying to think about, are there certain regions of the protein, certain regions of the channel that seem to have bigger issues? And if so, what are they associated with? Because if we could do anything to try and anticipate these issues, maybe we could do something to mitigate them or to make them less severe. So that was the reason for doing this. So I won't go too much into detail, but I listened to all of what you said in terms of the different symptoms, and I went through and I scored. And I said, well, for some things, for instance, developmental delay or hemiplegic migraines or autism or status epilepticus, we'll give a point for that. For seizures, depending on how many seizures a person has had, we'll give 1 or 2 points for that. Depending on how developmental milestones were coming, in particular walking and talking, we'll give some variable numbers of points for that, all with the idea of being able to, in the aggregate, get some sense of how the brain was functioning, and realizing that it's limited, but we wanted to get an overall idea. When we plotted that out in terms of seeing the distribution, we could see that with that scoring system, we could get a sort of reasonable distribution. We could spread people out in the community. And then what we tried to do was to look at the different types of genetic variants. And again, the numbers are small, so we couldn't look at every single variant uniquely, but we started to think about, are there particular regions or particular types that seem to be acting differently? And so this is how, using that score, that numerical summary score. And again, a higher number means more severe, a lower number means less severe. So if you recall, there was a type of genetic variant, it was in gray in that slide back a couple ago, where we thought that those particular variants might result in half of the amount of the protein being produced. And so we call those generally truncating variants for short. Again, when we think about this, we have one that's working perfectly fine, but the other one, it's as if it didn't show up for work. Those individuals tended to have the milder symptoms. So those tended to, in the severity score scale, they tended to be the lowest numbers. Now, if you'll recall, I also showed you those sort of squiggly lines going through, and I showed you some of the different S5, S6, or S4. And so if you break those down into those different regions, again, that are forming the core of the channel, we could start to see some general trends. But you will realize each one of these is a dot for a different person, that there is some variability within these broad groups. So not everyone, for instance, that's in a linker region or S3 is going to be exactly a 3.5 or a 4. There's some spread. Some people are 7, some people are 2. So there's a distribution around this. But on average with this group, they tended to have a lower severity score than on average the group that were at S4 or S5 and S6. I think there's a lot more we need to do to be able to get down to specificity in terms of specific function and specific amino acids, but it's to show that we're beginning to be able to do that type of analysis. Now, we also looked at things a slightly different way. Remember, I told you that we had being able to look at the channel when it's open too much, a gain of function, when it's closed too much, a loss of function, or in fact, as I said, with truncating. And again, we did this now not looking at the specific location, but looking at how the overall channel we thought would be working. And again, can see on average that when it's open too much, we seem to have more problems than when it's closed too much, and then versus when it's truncating. And again, though, there's still a distribution. Not everyone is exactly the same. And so there's some people with predicted gain of function variance, but they might have pretty good scores on comparison. And this may be in part that we have to look at the combination of the effect, plus in part the location, plus in part there are probably things that are more complex in terms of the way that channel functions. And so there's still a lot to learn, but we hope that by understanding this, we'll be able to, again, anticipate in part prognosis, and more importantly, I hope, come down to eventually what treatments might be helpful. So to that end, we have been asking you some really annoying questions, and I realize they are so annoying. But the annoying questions we've been asking are about treatment. And the reason we've been asking about them is because we wanted to know, is there something that looks like it works? Could we be able to put together what are best practices, if you will, or standards of care or clinical guidelines? I will say we are not there yet. Our numbers are not big enough. The distribution, the breadth of what we see is too wide to be able to come up with good data to be able to guide our treatment perfectly. But this is still the goal, and I'm sure we're going to get there. So we did ask, though, in terms of the medications for seizures in particular, because we heard you in terms of the concern, in terms of the burden, in terms of the impact. For some individuals, seizures are where it's at. So we were asking you what medications seem to work. Number one, many people had to take more than one medication. And at times over the life course, sometimes it was needed to change the medication. So I'm not saying that this is a simple, you just pick one of these and you're done. And this is something you definitely need to be talking with your neurologist about, with your epilepsy specialist about, because this can be something, again, that you will need to customize based on your particular child or family member. But I will say that what I learned from this is that it wasn't simply one size fits all. It wasn't as if there was one medication that was the magic bullet. As I said, in some cases, it requires 2 or more medications. And so in doing this, this is part of where the art of medicine comes in terms of being able to have a good doctor who's got experience and a gestalt about whether or not the seizures are under control, and also balancing the side effect of these medications, because some of you, as you know, are balancing the cost and the benefit, if you will, some of the side effects and the drowsiness that comes. In addition, obviously, it's not only about the seizures. Part of it is about the ataxia as well. For some individuals who have spasticity or problems with hypertonia, oftentimes this is something that evolves over time. Botox injections were helpful in terms of obviously sometimes needing to be repeated. And for many individuals also having, I call it tummy trouble, gastrointestinal issues. For some people, it's constipation. Other people, it's diarrhea or reflux. Some people, it's both. And on different days, it could be different things. I actually think these intestinal problems are related to what's going on in terms of the brain. I believe I know there are nerves that come from the brain ultimately to the intestine. And I think that's the connection in terms of those connections. Again, people have been using, in some cases, medication, in some cases, diet, but I know that that's something that oftentimes, even for nonverbal children especially can be quite irritable and upsetting, and sometimes you don't know why. I will say that there were some things we also heard in terms of downsides. So medications that can cause problems, and it's not everyone, but we did hear from certain individuals that propofol, a medication that we sometimes use for surgery in particular, was causing issues in some individuals, and it's something we need to continue to watch and pay attention to going forward. So as we're thinking about going forward, I do want to encourage, as you've been hearing today, we would like to be able to make this easy for you to be able to participate so that your voice is heard and that you're represented. And I do want to underscore this, especially for the onesies. If you recognized yourself as a onesie, or you know your child's genetic variation and it is not represented up on that chart, I don't want you to be left behind. And you may be the only person to be able to represent yourself. That is, that these individual genetic variations, to have the researchers work for you and help you to be able to understand your child's condition, you need to be represented. And that's why we're trying to make this easy for everyone to be able to participate. So if you're having trouble, if you get stuck, I do want to say that we've got help on the way. I'll get to this in just a second in terms of how you can get that help, but we do have researchers available. I apologize, Sean Calamia, who was planning on being there, had last minute change that he couldn't be able to be there, but he's there remotely with you for anyone that needs to have a Zoom visit with him to help you out. With doing this, as I said, this is not one and done. We have to understand how things are changing over time, what things are getting better, if they're getting better, what it is that you figured out to make them better. And then we also have something called a Vineland Adaptive Behavioral Scale, something about development of your child over time as they gain skills, and be able to monitor how that's happening, how quickly it's happening, what they're able to do over time. And with this, as we find out new questions, in part questions you raise, we will continue adding to the interviews that we're doing to drill down on the things that are important to you. So we'd love to hear feedback, things that are either a waste of your time, things that we didn't get quite right yet, or things that we're missing that we need to add. So with that, I want to thank everyone who's been involved, most importantly, the foundation and you all who have participated so far, the study team members from Columbia, Sean and Alexa as the coordinators who are happy and ready to help you. And I again put up one more time their email addresses so that if you do need help either getting started or getting stuck along the way, help is on the way. Okay, with that, I'll stop and I'll be glad to take questions.
Hello. All right, we're going to start with a question from the virtual audience, please. Somebody asked if you could clarify if gain of function are more severe. And then secondly, for those who reported successful treatment with Diamox, were those both gain of function and loss of function variants?
Good question. So yes, I should have said this earlier, so I'm glad you asked. In general, gain of function mutations did tend to be more severe. And if you happen to know your genetic variation, there was also one specific missense variant at position, depending on how you look at this, it's either 1392 or 1393, but one that we saw quite frequently, and that was also specifically associated with greater problems. And so I'm just showing this again. I'm glad you put that up. So this particular genetic variation that we saw over and over again. So both of those tended to be gain of function in that one specifically, more severe epilepsy in particular. With the Diamox, yes, we did. I have to go back and double check this to make absolutely sure, because I want to make sure to say that it wasn't only gain of function. It definitely was more gain of function, but I will get back to Lisa, who can get back to the group, just so I can be sure there weren't any loss of function variants as well.
Do we have any questions in the room? I'll come over there next.
Thank you, Dr. Chung. I have a quick question. In the natural history questionnaire, the way it's set up, do the participants have a chance to indicate the earliest symptom that they had, and also whether they had a chance to describe their most bothersome or debilitating symptoms?
That's a great question. So, in fact, it's not asked in exactly that way. And if the group thinks that would be helpful, we can certainly make that modification and add that. And it is something that I've been thinking about. So let me say a couple things. Number one is that part of the reason I do the natural history in general is that when we think towards the day of clinical trials, it's very important when we go to make the study design and when the FDA gives us permission to do clinical trials, it's very important to know how the condition evolves over time and, understanding the distribution, what the variability is between individuals. Now, the reason I mention this is because as we're designing that, I always ask families, I ask individuals, and I ask parents the question you asked, which is what matters to you? If we had to look at an outcome measure to say what would make a meaningful difference in your life, what would that be? Now, the feedback that we've gotten, and again, it's variable depending on what symptoms the individual is having, but the feedback we've gotten is largely that it's related to the epilepsy. And if people differ, that may be because not everyone has epilepsy, but that's the feedback that we've seen so far. So we've been thinking ahead in terms of doing that, ways that epilepsy groups in general think about how to monitor seizures, very fancy ways to do this, both in the home as well as the way we do these in hospitals. So if folks have differing opinions or if they think that's not quite right, there's something else we should be querying, I'd love to hear about it, but that's what we've heard so far.
All right, thank you, Dr. Chung. I have another question from a family out here, and I think this is probably something that many families are wondering. Can you give us sort of a general idea about how gain of function and loss of function are predicted? Are you going based on just symptoms, or is there other work that's being done to classify them?
Yep. So I am going to give a preview, but I'm going to say that you've got many brilliant scientists around the room and who are going to be speaking at the conference. But this goes in the following way. So folks like Al George do very elegant studies where they can do patch clamp experiments to actually look at individual ion channels and to see how they work, whether they stay open, how long they stay open, what the voltage sensitivity is, very fancy things to see how those work. This is something that's now done with data science in general, is that you can gather up a lot of information from people like Dr. George and others and see how those channels work. And you can start to see patterns and make predictions. And so we can use things called machine learning and artificial intelligence to be able to make predictions. To answer your question, no, it's not based on symptoms of the individual person. When they report to us they have epilepsy or they have problems in terms of development, it's based on the electrophysiology of that individual channel and making predictions based on, again, pattern recognition essentially is what it comes down to. Now, it's not perfect, and it's not perfect because, in fact, we haven't saturated this. We don't know every single amino acid in the protein that can be changed and how it's changed. So these things get iteratively better the more data you put into them, the better we are predicting. But that's the science of how that's done.
Another question from our audience.
Hi, thank you. I'm wondering how you factored in or what difference it makes if it's specifically a stop variant in where that stop occurs, or if that has something that's factored in or how that works.
Okay, very good. So this is a great question, a nuanced question, and I'm going to explain it, although if you'd like to talk with me offline, I'm certainly happy to. So for the rest of the audience, there are certain genetic variants, and in the picture that I showed you, they have these little stars or what looks like an asterisk. Any of those were leading to what we call, instead of making a full version of the protein, it goes part way and then it stops. And so we call that truncation. That's just a terminology that we use for that. We assume that in most cases, the cell will actually take that version of the protein and kind of send it to the garbage and recycle it and not use it. And that's an oversimplification in the analysis that I showed you. We did not actually test that to know that that was the case, but we assumed that that was going to be the case for enough of the different premature stop mutations to think that that would be how it would work. Now, if you're really thinking about this carefully, you'll realize that that may be an incorrect assumption. There are things called alternative splice variants. There are things that are at the way end of the protein where it still might make a protein, and especially when something is in what we call the last exon, that becomes an issue. For the variants that I showed you, those were not predicted to be issues. That is, that these were in splice isoforms that we thought the brain was using, and I didn't include things that we thought did not lead to what we call nonsense mediated decay. So that's sort of biology where it's the amount of the busted.
Dr. Chung, can you still hear us?
I can hear you now.
Okay, our connection was unstable, but it looks like it's better now. Do we have any more questions from in the room? Okay, perfect. One moment.
You show some medication that some patients respond to them and some do not. Do you know if the patient actually responding to the medication they are because of some placebo effect or not? Is there any comparison for that?
So there is, to answer your question, no. So this was not a controlled study. This was not a clinical trial. This is not as if we put everyone on one or put half of the people on the medication, half of the people on a sugar pill or placebo and see who responded. It was not anything like that at all. This was simply people and their doctors going throughout usual clinical care and then reporting back to us what they observed. So this was not, from a scientific rigor point of view, not like a well-controlled study to be able to do. But on the other hand, this is not something diagnosis when they started the medication. So this is opportunistic. I totally grant that in terms of being able to get the information that is already available. But at some point, I anticipate there will be clinical trials that will be quite rigorous and that will go from, for instance, what your usual medication is to, for instance, being able to be compared to a new medication. And in those cases, I do expect, whether it's a placebo or something else, that they will be very well controlled.
Thank you, Dr. Chung. I have a question from a parent who has a son who's autistic. She was wondering if she would like to know the correlation between autism and CACNA1A, and if there have been any studies with aminopyridines regarding that.
Very good question. I think about this a lot because I think about both CACNA1A and I think about autism, and they're absolutely connected. This is not to say that everyone with CACNA1A has autism, and this is not to say that everyone with autism has CACNA1A, right? There's an overlap in terms of the two. I'll say something that I hope will make sense. Autism is a behavioral diagnosis. So by that, I mean, it's a description of certain behaviors an individual has that are related to everything from how they interact with other people to some oftentimes repetitive behaviors or a need for consistency and sameness. And that's in sort of one dimension as we come up with an autism diagnosis. CACNA1A is a genetic diagnosis and can envelop many different manifestations, including autism, developmental delay, epilepsy, hemiplegic migraines, everything we've been talking about. So I consider those kind of orthogonal. That is, they're two different dimensions and sometimes they overlap in terms of that. When we think about the medications that might help the autistic behaviors in general, although this is pure speculation, so let me be clear, pure speculation at this point, I think in general, things that help the brain for CACNA1A, so things that reduce seizures, things that reduce ataxic episodes, but things that help the brain function better, I think probably help globally in terms of cognition, in terms of autistic behaviors. I think they sort of leach out and help other things as well. So within that, I think you get kind of global benefit if you can find something that's helping.
Question from our live audience.
Hi, Doctor. Based on the number of participants you have in the natural history study today, I'm just wondering if you have a view on how frequently you sort of publish new research?
Okay. So I guess there are two ways to think about this. So I think there are publications and peer review where it goes through a laborious process in a good way to make sure that what we publish, other scientists have had a chance to improve it or to criticize it if they think there's something wrong with it. In terms of doing that, I would hope that we'd have new information for the community to be able to do that. It's a lot of work, and we want to make sure there's something, a new story to tell, so to speak, in terms of that. There's not a specific number that we come to with that, but it's more based on, I think, new good information for the community. On the other hand, there are other ways of quote unquote getting the information out. I'll say number one, hopefully you appreciate anyone in this study is their privacy is protected. We don't put anyone's name on this. We don't identify anyone in the process. And you're going to be hearing throughout the meeting other ways that we share. Number one, all of this information, all of the researchers are able to use it. We don't keep this to ourselves. Again, it's de-identified, so no one knows who you are, but we enable all of the community, all of the research community to be able to benefit from the information that you're giving. We don't sort of own the data in that way. Because of that, there are many people who are continuing to think about and learn about this information, and they will publish, I think, with the many different things that they are learning as well. So aside from anything with what I've shown you today, they might do things that are looking at how the ion channels are functioning, or they might look at other calcium channels in comparison to this. So I think there are lots of things I can't even imagine. I'm not creative enough to think of everything that they will do, or publications will be coming out of this as well, different dimensions that come out. It is also in terms of coming back to you all, this is really your data. So when you think about this, I'm happy, to hear from you with what periodicity you'd like to hear about updates. And I think the good one we've been on the cadence to do is an annual update. If you all feel that there's some other way of doing things, I'm open to suggestions, but at the very least, I hope we'll be able to give the community an annual update.
Oh yeah, we have another question from a parent online. She's asking, Dr. Chung, have you seen any patients who are taking antipsychotic drugs for CACNA1A, or any, young girls who need hormone therapy?
Tell me a little bit more about the hormone therapy for the young ladies.
I can ask
that
the
question
parent
again.
All she asked was if you know of any girls who need hormone therapy. I would assume in response to maybe I think that there are thoughts that sometimes some of the episodes can get wonky, or symptoms get wonky during puberty. So maybe she's talking about that. I'm getting some nods from parents here.
Yeah, so I guess the way I'm interpreting it, although you'll tell me if you think differently, it's definitely, so one of the things I didn't explicitly say, but the reason we do this over the life course is, like you said, puberty is definitely a time when things change. And I should have said that explicitly. So when the hormones change, things can change. And I have to tell you, I have teenagers. So, normal teenagers, things change and things get wonky. So let's just call that the baseline. But in addition, with our community, whether it's epilepsy or other things can change as well. What I have seen is that there are individuals who will work with a gynecologist, oftentimes an adolescent gynecologist, and there are things that I won't call them hormone replacement, I'll call it hormone regulation, I think is what the question is really about. And yes, there are individuals who are on some sort of hormonal regulation, whether it's an injection, whether it's oral contraceptives, but other things to be able to help with cycles, and in some cases, both make periods less painful or have lower flow, as well as sometimes have an effect in terms of behaviors or epilepsy. So, but those are, again, talk to your doctor in terms of any of those specifics. With the antipsychotics, I'd have to go back in terms of exactly the age of the individuals used. It's not a common thing to take, but there are several different medications that folks have used for different types of behaviors. So not common, but yes.
Okay. And do we have time for another question? Okay. Just a really quick one here that I think, some parents might be wondering before they enroll in the study. A parent asked that if we enter or enroll our son in the study, will his genetic code or DNA end up being patented during or after the research?
So with this, actually from work that I had done, genes aren't patented. So we consider these products of nature, and you don't own them, so that it won't get patented in that way. So no, nothing I don't think is going to be patented that comes out as a result of this. I will say that if you don't get represented within the research, you run the risk of having a treatment that might be developed that'll work for someone else but won't work for you. So you've got incredibly brilliant, hardworking teams of people working on this, teams of scientists around the world, and especially around the United States. So if it were me and my child, I would have them work for me. So I would, whether it's my registry or you just go up to them, however you want to do it, but make sure that you're represented.
All right, thank you. Allison, if there's anybody else.
Dr. Chung, this is Sunitha with the foundation. Thank you again for your time today and just all of your dedication to this community and doing this natural history study. Question for just the community. You have, I think you said 89 individuals enrolled, and you expect this to be an quote unquote equal opportunity disorder. So there's clearly, we need our numbers to go up to really start to tease out some of these trends. Like is this in fact something that is appearing more frequently in females, for example? What is that target number? What should we be shooting for? Obviously, we want every single patient to be represented in the study, but short term, at what point can you really start to say, okay, this is accurate because we have X number of individuals enrolled?
So what I'd say is I'm worried because, as I showed you on the picture, most of our genetic variants are onesies at this point. And so that's always hard to know, it's based on just one person, how accurate we are within that. I'm not going to say an exact number, but I'll also say that the larger our numbers are and the more that we can show companies that are developing treatments that we are well organized, that we have a registry, the more that they see a market. I'm just being really crass about this. They see a market to work towards for a treatment. And so they do go and they try and decide, should I work on this, or this? And they go by the numbers. It's a numbers game for them. So the more that we can show that we're organized, we have large numbers of individuals, we have the resources, we've met them halfway with knowing the natural history, potentially having cells that they can work on to represent our children and ourselves, we de-risk it for them. We make it easy for them. And then it's like, I think of it as flowers for honeybees. We make it attractive for them and come to visit us and to work with us when they could have other opportunities. So I'm going to throw out a bold statement. When can we get to 500? Can we make that a goal? And I know it's audacious. I know it's a big number, but I think when you start getting to those, people start paying more attention.
Thank you, Dr. Chung. And with that, we'll just ask everybody who hasn't already completed the natural history study to do it. If you don't stay up late tonight and do it another night soon, so we can get to 500. Thank you for your time, Dr. Chung.
Thank you.